Hello everyone, today we are going to learn about xenobiotics. So, we will begin with some introductory points, right. So, what are xenobiotics? Xenobiotics are compounds which are basically foreign to the body. So, some examples of xenobiotics, they can be drugs, they can be chemical carcinogens, they can be plant foods, they can be polychlorinated biphenyls, that is PCBs, they can be insecticides, they can be pesticides, pesticide poisoning and all those pesticides, etc., right. So, obviously they need to be metabolized before being excreted. So, what is being metabolized? That is detoxified, right, detoxified, or in some cases activated before being excreted, right. Where does it happen? Mainly in the liver, okay, mainly in the liver. So, now let us move on to the metabolism of xenobiotics. So, now let us talk about metabolism. Okay. It happens in two phases. So, first let me briefly explain you about the two phases. For example, X is a xenobiotic, right. What happens in phase 1? Okay. We add something, for example, Y. Let me explain it in very layman term, right. In the beginning, we add Y, it becomes X Y. In phase 2, we add Z, for example, we get X Y Z. X being our xenobiotic. So, this was our phase 1 where we added Y. So, what happened is that phase 1 metabolism rendered the compound more reactive, introducing groups, that is Y, that can be conjugated, that is in phase 2 conjugation or acetylation or sulfation, whatever, in phase 2, that is Z here, with glucuronic acid, sulfate, acetate, etc. Basically, why we are doing this? Let us understand the reason why. We want to make it hydrophilic. Why we want to make it hydrophilic? So that we can excrete it. Excrete where? Excrete in the urine, excrete in the bile. So, if X is a xenobiotic, we added or we reacted in phase 1 of the metabolism, after which it became a little bit more hydrophilic. Therefore, this Y group can now attach with several other compounds, several other groups like sulfates, acetate, which will make it more hydrophilic. Therefore, it will be excreted in urine. So, this is basically what happens. There is phase 1, we have our xenobiotic, Y for example is being added, then phase 2 where conjugation usually happens, conjugation with glucuronic acid, sulfate, acetate, glutathione, amino acids, etc., which we will learn in the later slides. Okay. So, this is the basic, you will understand as we proceed. Okay. Now, what happens? What are these phase 1 reactions? First, let us know them. So, they are hydroxylation, right. Hydroxylation catalyzed by what? We need enzymes. So, hydroxylation by monooxygenases or your mixed function oxidases, right. Monooxygenases genases, right, or your cytochrome P450, right. Hydrolysis reactions can happen catalyzed by esterases, right. Your there might be some non P450 catalyzed reactions as well, right. You know, there might be an additional phase 1 reaction. What do I mean by additional phase 1 reaction? For example, we have something like pro-carcinogen, right. We are having phase 1. Then if it becomes biologically active after phase 1, that means what? It becomes harmful, right? Carcinogenic, not good for the body. So we will have here additional phase 1 reaction, which will make the compound less active, or I can say inactive, therefore harmless or less harmful, you can say. Then we will go for conjugation, that is phase 2, usually conjugation. Then we will excrete it, excretion. So there is intake, there is metabolism, and there is excretion, as simple as that. So the hero here is cytochrome P450. That's the protein or enzyme that I should say is catalyzing most of these, phase 1 reactions, cytochrome P450. So let's learn about this, okay. So cytochrome P450, first of all, is a heme containing enzyme, right, and there are 57 cytochrome P450 genes in human genome. Some facts given in Harper, okay. So why suddenly P450, right? This was because this cytochrome P450 were found in microsomes. What are microsomes? These are basically ER fragments, right. Now these ER fragments, when they were chemically reduced and then when they were exposed with carbon, or rather exposed to carbon monoxide, then their absorption peak was at 450 nanometers. Hence the name cytochrome P450, right. Obviously microsomes contain cytochrome P450, right. ER fragments, it is present in SCR membranes. Now whatever. Now basically what this cytochrome P450 does, what is the reaction? So first let us write the reaction, phase 1 reaction, right. Let us take an example. For example, this RH. What is this RH? This is a xenobiotic. So let me first write down the reaction, then I will explain you everything, okay. RH plus oxygen, we need NADPH plus H plus here. This will go to ROH plus H2O. Next function oxidase, NADP. Here we have cytochrome P450, it's getting reduced, cytochrome P450 reduced form, right? Let's write the two enzymes. The two enzymes will be monooxygenases. Let's write it MO here, okay, and here it will be NADPH cytochrome P450 reductase. Okay, what basically happens is, see what we are doing here. This is a xenobiotic. We are making it a little bit more hydrophilic so that it helps in conjugation, that is phase 2, and which in turn will help in its excretion, right. So, this is ROH is being formed, right. So, we need cytochrome P450 here, we need monooxygenases here, we need this NADPH cytochrome P450 reductase enzyme here. So, what is happening, this enzyme NADPH cytochrome P450 reductase, this enzyme is helping or catalyzing the transfer of electrons from where? From NADPH to cytochrome P450. So, this is getting reduced. Now, what this cytochrome P450 is doing is, this reduced cytochrome P450 catalyzes the reductive activation of molecular oxygen, right. One atom of which becomes the hydroxyl group in the substrate and the other is reduced to water. Obviously, it is not dioxygenase, it is monooxygenase. So, hence the enzyme. So, this was our substrate xenobiotic, this is our product ROH, right. This was the phase 1 reaction. After ROH, I mean on ROH, we will have many more phase 2 reactions and then we will excrete it. That is the basic concept here. Okay. So, there is another cytochrome since mentioned, let us talk about it. It is cytochrome B5, right. It is again a hemoprotein. It is present in the SER membrane, and here it acts as an electron donor in some cases, sometimes. Okay, done. Let us move on to our next topic or subtopic of systematic nomenclature. So, systematic nomenclature of what? Of cytochrome P450. So, remember, we are naming cytochrome P450 to be very specific, not any other cytochrome, cytochrome P450. So, let us take an example, it will be easier for you. Okay. So, let us take an example of CYP1A1, right. What is this? This is a cytochrome P450, its root. What is its root? This CYP's root. This signifies cytochrome P450. This we call is the root. Okay. So, this is a cytochrome P450. We are naming it, right? Just like enzyme commission code, EC code 1111. As far as remember, it was alcohol dehydrogenase or something. I don't remember. Whatever. Yeah, oxidoreductase. So, 1 it should be, whatever. So, like that, you know. So, this is what? This one, this is family. That means same family, right? This capital A is what? Subfamily, and this one is what? This is not subfamily, it's individual. We can say that this is the first individual member of that subfamily, right? So two cytochrome P450 will fall in the same family if they have greater than equal to 40% amino acid sequence similarity, right? I'm just writing greater than equal to 40% amino acid similarity. Let this be the sign of similarity, okay? And here subfamily greater than equal to 55% if they have amino acid sequence similarity, then they will fall in the same subfamily. And obviously individual member, it's totally unique. So yeah, so this is the enzyme, this is the protein. Now gene encoding this cytochrome P450 will be written in italics, just that, CYP1A1. This is in italics, right? This is nothing but gene encoding this protein, encoding this enzyme, encoding CYP1A1. Simple. Okay. Now let's talk about the major cytochrome P450 involved in drug metabolism. So drug metabolism we have CYP1, CYP2. These are what? These are families of cytochrome P450, right? CYP3. These are the three families which are mainly involved in drug metabolism. Cytochrome P450 or CYP1 has 3 subfamilies. CYP2 has 13 subfamilies. Like you can say CYP2E, CYP2D, CYP1A, like that, right? 13 subfamilies. CYP3 has only 1 subfamily. Okay. Obviously they have, these are what? Cytochrome, these are enzymes, they'll work on substrates, right? So do they, do they react with unique substrates? Do all of them react with unique substrates? Obviously not. They have overlapping substrate specificities, right? Okay, next point. Where are they found? They are found in liver cells mainly and enterocytes. What are enterocytes? Enterocytes are basically intestinal cell lining cells, right? So they are where? They are SER membrane. We call this what? We call this microsomal fraction, as said before, when a tissue is subjected to subcellular, we can say, during subcellular fractionation, right? This is it. You will be surprised to know that cytochrome P450 accounts for sometimes 20% of total protein in hepatic microsomes. it is 1/5, you know. Then in adrenal glands, these are some, you know, facts given in Harper again. In adrenal glands, cytochrome P450 is also present, obviously for cholesterol and steroid hormone biosynthesis. This BS is biosynthesis, nothing else. In mitochondria mainly, and ER, right? One more thing which you need to know is that most isoforms of cytochrome P450 are inducible. They can be induced. Example, phenobarbital. What is phenobarbital? It's a drug, right? It's a anti-seizure med or anti-anxiety med, anti-anxiety medication, right? Okay, this phenobarbital, this causes SER hypertrophy. This leads to 3 to 4 times increase in cytochrome P450. Therefore, you know, since obviously cytochrome P450 is there in SER membranes, hypertrophy is there, so obviously their size, their quantity will increase. Now, what's the reason? The reason in most cases is due to you know, increased transcription, right? Most cases it's increased transcription. However, in some cases this induction involves stabilization of mRNA or the enzyme protein itself. So increased transcription is the main cause. Some other minor causes may be mRNA or protein stabilization and increased translation of existing mRNA. Okay, so yeah, these are the this can be an example. So, you know, now let's learn about various drug interactions, right? Induction of cytochrome P450, this underlies drug interactions, like when the effects of one drug is altered by administration of another. Let me explain it, okay, with the same example. Phenobarbital, right? This induces what, you know? This induces CYP2C9, right? And what does this CYP2C9 does? It metabolizes warfarin. What is a warfarin? Warfarin is an anticoagulant, right? Therefore what? If a person is taking an anti-seizure and an anticoagulant med together for whatever reasons, right? Phenobarbital and warfarin, if the person is taking together, for example, in this case, what will happen? Phenobarbital will induce CYP2C9, which helps in metabolization of warfarin. Therefore, warfarin will metabolize faster. Therefore, warfarin will remain in the blood and the system for lesser time. So, it will reduce efficacy of what? Of warfarin. So we need to do what? We need to increase the dosage of what? Of warfarin. So warfarin dosage of a person who is taking phenobarbital, for the same outcome it will be more than a person who is not taking phenobarbital, right? This was one example of drug interactions. Another example, ethanol again induces what? CYP2E1, right? This helps in the metabolism of procarcinogens in what? In tobacco smoke. Many people take both, obviously, but you know, it increases risk of carcinogenicity, therefore quite obvious, right? CYP2E1 metabolizes procarcinogens in tobacco smoke. Ethanol, if you take ethanol while you are having cigarettes, it induces this CYP2E1 and procarcinogenic metabolism increases, so carcinogenicity increases, quite obvious. Okay. Another thing, you know, I might have seen in some prescriptions there written that do not take hing, or you know, do not take grapefruit or something with these medications, written in all the prescriptions. I guess I have seen in homeopathic prescriptions or somewhere. I do not believe in homeopathy, but still many do. So you know, I think that's the reason for which we are discussing now. Grapefruit. Grapefruit contains what? Furanocoumarines, right? These, what do they do? These suckers right here, these inhibit cytochrome P450. So therefore, the drugs which were stimulated by cytochrome P450, they will be inhibited, and those that were inhibited by cytochrome P450, they will be induced, right? So just the opposite. So not going into enough details, what's the drug metabolism which are affected, you know, in this case are the metabolism of statins, omeprazoles, antihistamines, that is anti-allergic meds, benzodiazepines, antidepressants. Right, antidepressants. Now another thing which you need to know is that polymorphism of cytochrome P450 is responsible for variation in drug responses by different patients. Now what do I mean by that? See, for that you need to know what is polymorphism, right? What is polymorphism? We have learned that in, you know, class 12 in the chapter of molecular biology or something. It was given in NCERT. Polymorphism is nothing but it refers to the presence of two or more variant forms of a specific DNA sequence. This occurs in individuals or populations, and the most common one is SNPs, right? Single nucleotide polymorphism. So, if there is a variant with low catalytic activity, right? So, there will be what? Cytochrome P450, low catalytic activity. There will be low or lowered action of cytochrome P450, therefore slow metabolism, therefore prolonged drug action, right? Opposite, high catalytic activity, faster metabolism, decrease in efficacy or decreased drug action. Everything is just the same, you know. We saw it here. Phenobarbital metabolizes warfarin, increase in dosage, reduce efficacy. The concept is the same, everything is just the same. High catalytic activity, faster metabolism, decreased efficacy. Less in blood, remains for lesser duration in the system. It's all the same. Now there's one more thing, that is nicotine. It's metabolized by CYP2A6 into conatin, right? Now the CYP2A6, let me tell you, has 3 alleles, one wild plus 2 inactive. Now if a person has these 2 inactive types, like if he has inactive alleles present, then what will happen? CYP2A6 will be inactive, nicotine will, you know, comparatively remain for a longer duration in the system, in the blood, in the brain. Concentrations of nicotine will remain elevated longer than those of individuals with the wild type allele. Again, same concept, same, nothing different. So this you know, inactive allele, I can say this protects smokers from being, or protects people from being tobacco dependent smokers, right? You know, nowadays novel therapy is being applied. That is what we do, we inhibit this wild allele, we inhibit this with the help of some inhibitor. So therefore nicotine will stay for a longer duration in the system. Obviously everything is way more complex than that. It may help in smoking cessation, right? Obviously the world will become a happy place, like your mama says. So this part 1 covers this only. In the part 2 we will learn about the phase 2. So we introduction, we learned about the introduction, what are these PCBs and all these things. Metabolized, where it happens, liver, 2 phases, right? Hydroxylation, hydrolysis, non P450. Additional phase 1, additional phase 1, like there will be an additional hydroxylation and all those things will happen. It's again a heme containing 57 CO, chemically reduced. Microsomes are ER fragments, absorption peak at 450 nanometers. We learned about these things here. Cytochrome B5 electron donor, and greater than 40, greater than 55, equal to, greater than equal to. So root, family, subfamily, individual member, gene in italics, whatever. CYP 1, 2, 3, 3, 13, and 1 subfamilies, overlapping substrate specificities, present in liver cells plus enterocytes, SCR membrane, microsomal fraction during subcellular fractionation, cytochrome P450 20%, adrenal glands in mitochondria and ER, cholesterol and steroid hormones. Obviously, that thing BS is different. Are inducible. We talked about phenobarbital and everything. Again, CYP2C9, 2C9, phenobarbital, ethanol, 2E1. We talked about grapefruit, furanocoumarines. We talked about polymorphism. We talked about 2A6 and nicotine. So this is for part 1. Thank you for watching.